All right, so facilitated diffusion, it's another type of membrane transport, and similar to simple diffusion, it's still diffusion. Things move from high to low concentration, but remember the membrane, which I symbolize with the red line here, is not permeable to all compounds. And so if they're charged or polar, a little bit larger, they're going to need this special channel to move through, but they still move from high to low concentration. Let's take a little closer look. All right, so our membranes are a little bit like these window screens, right? Certain things can move through, but not everything can. So we put these in our windows so all the flies don't get in, right? But the air still moves through, cools down the house, creates breeze. Really nice, things like that. So it's selective. It's not completely impermeable, but only some things can get through. If we wanted some things to get through, then we'd need to cut a little hole. And so that way, that could pop through, but we can seal this back up and still keep the flies out. So what would that look like in a cell? Let's take a look. So facilitated diffusion is also a passive process, meaning it doesn't take any work. It's just that Brownian motion, jostling these around in random motion, and they spread out from high to low concentration. When we looked at simple diffusion, we only considered the phospholipid bilayer, but now we're going to look at the whole mosaic, at least some of the parts like these protein channels over here. So let's look at a simplified model of that. So some of those protein channels, this is a protein that the cell made and installed in the membrane, so it just sits there, and this one is shaped for specific ions. Maybe that's a sodium ion, maybe it's for a slightly larger potassium ion, or even a chloride ion. But each of these channels is built pretty specifically for one specific type of ion. It doesn't tend to let others through. There's a little wiggle room there, but for the most part, you can think of them as very specific. And this is just facilitated diffusion, right? So things are moving from high concentration, like you see those orange positive ions there, to low concentration, like they are down here. So the net movement is going to be across this way. None of these ions can move through the phospholipid bilayer, so they're going to have to use this protein channel instead. It has this permanent hole in it, but it's just big enough for those ions to get through and really nothing else. So it's not compromising the integrity of the cell. So it would look something like this. And now the concentrations are even, and the net movement stops. Now it could be that this was the outside of the cell and those ions were moving in, or it was the inside of the cell and the ions were moving out. It doesn't really matter. It can go either way. The ions are just going to move down their concentration gradients. Well, that's ions, and the channels. If it's a larger molecule, we need a slightly different configuration like this carrier protein. So something like glucose is much larger, but it still has a high concentration and a low concentration. So those are going to move into the cell, and let's watch as this glucose moves through. Now we can't create a big old hole like this channel, or it would ruin the integrity of the cell. So we have this protein that changes configuration. Once that glucose sits in there, it shifts, dumps the glucose out, and then goes back so that it can receive another glucose. It will keep doing that until concentrations are equal. Now, one application in the cell here is that as this cell uses up glucose, the concentration tends to stay fairly low inside the cell. And so glucose usually stays pretty steady outside the cell because we maintain our blood sugar. And so there's usually this kind of steady flow of glucose through these carrier proteins into the cell. And like that flow down gradient concept, this membrane represents resistance. There's more resistance to flow because these can't pass through. They kind of bounce off that membrane because they're polar or charged in some way. So they can only move through here. But the more of these we make, the less resistance there is, and the faster these particles can spread out. Cells can control how quickly particles spread out by controlling what proteins they put in the membrane. So a cell controls its protein production, and the more proteins it puts in, the less resistance. So if it wants a lot of a substance to come in or leave, it makes more proteins. If it wants to be more of a barrier, then it makes very few. For example, sodium. Usually the cells don't want much sodium inside, so they produce very few sodium ion channels. This is the core of homeostasis, even at a cellular level, controlling how much of each protein it makes. So there's facilitated diffusion. Don't get thrown off by the word facilitated. It doesn't mean work. It just means we're opening little windows to facilitate diffusion. It's still a passive process. It just applies to ions and some of those polar molecules like glucose and amino acids. It's an important process and one of the important ways that the cell controls its internal environment, keeping it different from the outside. And that's a key property of all living things. It's homeostasis at its core. Well, that's it for this time. Until next time, keep appreciating life, even the little things.